The inaugural professorial lecture is the pinnacle of an academic in a university setting, so it's great to see so many friendly faces, people who have known Michèle for a long time, colleagues, students and friends to share in this evening with her and with us. Professor Prinsep is a Professor of Chemistry and the Team Leader of Chemistry and Applied Physics here at the University of Waikato. She earned her Bachelor of Science Honours and PhD from the University of Canterbury, where she began her research into natural products by studying marine invertebrates. She then undertook postdoctoral research at the University of Hawaii, focusing on cyanobacteria before returning to New Zealand to join the University of Waikato. Her research centres on the isolation and structural elucidation of natural products from an extraordinary range of environments, from Antarctica to the Pacific Islands, and organisms including marine invertebrates, cyanobacteria, fungi, algae, and plants. Her work is both highly collaborative and multidisciplinary, and in recent years has addressed nationally and internationally significant issues such as crop pathogens, invasive marine species, and food and water safety. Michèle is also deeply interested in chemical ecology, looking at the role natural products play in the everyday lives of organisms, both to better understand ecosystems and to explore potential applications of these compounds for human benefit. Professor Prinsep has attracted over $3.5 million in external research funding, and while much of her work is commercially sensitive and cannot be widely shared, her scholarly output is nonetheless prolific. Professor Prinsep has more than 100 published research outputs, including journal articles, technical reports, and book chapters, and over 90 national and international presentations, with many as a plenary or as a keynote speaker. She is a long-standing author of the annual Marine Natural Products Review, published in Natural Product Reports, an internationally recognised and widely consulted resource. Her sustained excellence in research has earned her a place on the Clarivat highly cited researchers list every year since 2015. Beyond her research, Professor Prinsep is a dedicated contributor to the wider scientific community. She is a committee member of the Australia-New Zealand Marine Biotechnology Society, a member of the International Bryozoology Association, and a fellow and former president of the New Zealand Institute of Chemistry. She currently chairs the NZIC Chemical Education Trust and has served on its National Council for 15 years. She is also active in peer reviews, postgraduate thesis examinations, and is an editorial board member of Marine Drugs. In addition to these achievements, Professor Prinsep is a passionate educator and advocate for science engagement. She is particularly involved in outreach with secondary schools and was awarded the New Zealand Institute of Chemistry's Chemical Education Prize in 2024. It is now my great pleasure to invite Professor Michèle Prinsep to the stage tonight to deliver her inaugural professorial lecture. Please, a warm welcome.
Tēnā koe, Sarah Jane, and thank you very much, everyone, for coming this evening. Tonight, I hope to show you how fascinating natural products are and why they're important to all of us through giving you an overview of some of my research involving them. Firstly, what are they? Well, you might think that's obvious. A natural product is anything obtained from a natural source, but that's not the definition that chemists use. To a chemist, natural products are synonymous with secondary metabolites. Unlike primary metabolites, which are essential for life, these secondary metabolites have very variable production. They can be influenced by all sorts of things, including environment and season. They're biosynthesized via specialized pathways, and they're often characteristic of certain species, genera, or strains. In many cases, we have absolutely no idea what their function in the producing organism is. Where we know what they do, I like to say that they improve the quality of life of the organism. So in other words, they help the organism thrive rather than just survive. Why would we be interested in them? Well, they have very unusual structures. Often they're very complex with unusual architecture. Basically, nature's got a lot more imagination than organic chemists have. For example, if we look at this molecule, it's a relatively small molecule, but it's got an awful lot of different functional groups packed into its structure. Natural products are involved in many biological interactions, and because they affect a living system, we describe them as being biologically active or bioactive. These bioactive natural products are often extremely potent because they have to be, because they're diluted by the medium in which they're transmitted, be that air or water. You've probably heard people say, oh, if it's natural, it's got to be good for you, but that couldn't be further from the truth. Natural products have both positive and negative effects. For example, Taxol is an extremely potent anti-cancer drug that's obtained from the bark of the Pacific yew tree, whereas botulinum toxin A, and yes, that is the one used in Botox, is the most toxic product known, and it is 15,000 times more toxic than the most toxic synthetic compound. If we're interested in these bioactive natural products, where should we look for them? Well, most of the biota on the Earth are concentrated into a pretty small area. So this gives rise to high population pressure, and it means that we have these very competitive and complex communities. So these are a really good place to start looking. An example of these are marine invertebrate communities. Marine invertebrate animals, such as sponges or tunicates or ascidians, are anchored to the seafloor. So they're competing for space and nutrients. They're soft-bodied, so they're vulnerable to predation. They're also vulnerable to overgrowth and microbial infection because they can't move. So it makes sense that if they lack an immune system or physical protection, that they might use chemistry to help them live better. Microorganisms are in a similar situation. They live in a complex soup of other microorganisms, so again, they're competing heavily for space and nutrients, so maybe they use chemistry to help them do this. How do we study these natural products? Well, to get enough of them to work on, for some microorganisms, we can culture them, such as these cyanobacteria here. But for other organisms, we have to collect them from the wild. We then extract them in a suitable solvent, and in the case of SpongeBob here, we make sponge smoothie. Now that we have our crude extract, and I have to say that was very satisfying, we then fractionate it using various forms of chromatography, ranging from simple bench columns to more sophisticated instrumental techniques. Once we have our natural products isolated, we have to determine their structures. We have a number of means available to us to do this, but the most useful by far to us is nuclear magnetic resonance, or NMR spectroscopy. This technique uses a very large magnet to interrogate certain atoms in our structure. As our natural products are organic molecules, we're interested in the spine of the molecule, which are the carbon atoms, and also the more peripheral elements, which are the hydrogens. And by examination of these, we derive a lot of very detailed structural information. The technology in an NMR spectrometer is the same as that used in diagnostic medicine in an MRI scanner, but our magnetic fields are much higher than that. We can also use mass spectrometry. This tells us about the size of our molecules and what type of atoms and how many of them are present. And if we manage to grow crystals of our molecule, we can use X-ray crystallography, or as I like to term it, cheating, because X-ray crystallography involves irradiation of our molecule with X-ray radiation, and then the diffraction pattern produced. From that, we can derive a three-dimensional picture of our molecule. So how did I get into this field? Well, as Sarah Jane said, I began my work on marine natural products in general during my PhD at the University of Canterbury under John Blunt and Murray Munro. They had relatively recently got a very large grant from the National Cancer Institute to look at anti-cancer compounds from marine invertebrates. This grant enabled them to employ a team of divers, including our very own Chris Battersell, 2 postdocs, and a technical assistant. And at any one time, they probably had 5 or 6 PhDs working for them, although they didn't fund us. Most of us, including myself, were funded on scholarships. They're a bit more abundant than they are today. So I got from honours to PhD with some work and some play, as you can see. Beyond that, I went on to do postdoctoral research at the University of Hawaii with Dick Moore. This was working on cyanobacteria, which are photosynthetic microorganisms, and this was a very well-funded group. In this photo, 6 of the 8 people are postdoctoral researchers, and there was a whole basement devoted to culturing the cyanobacteria on which we worked. Greg Patterson, who was at the back of that photograph, was able to employ quite a number of technical assistants, shown here, to both culture and extract the cyanobacteria for us. So that was a big group, and it was a nice, friendly group, very international. I now want to move on to some of the more specific areas that we've looked at, and starting with New Zealand marine bryozoans. These organisms look like plants, but they're actually animals. They have very primitive animals with no immune system. New Zealand's a really good place to work on them because we've got an excellent species diversity of bryozoans, and also we have a high proportion of endemic species, so that means the novelty factor is going to be higher. We've looked at all sorts of compounds from these organisms over the years, and this is a few of the families shown here. This work actually started with my very first PhD student, Bruce Morris. And I'm just going to focus on one of these groups of compounds, and that's the terrestrials, of which the first one isolated is shown here. These compounds were isolated from an endemic species, and they're potent anti-tumour agents. They kill cells by apoptosis, or programmed cell death, which is characterised by the membrane blebbing that you see in this picture. It appears that they target the mitochondria, or the energy factories of the cells, and we can see this if we look at the image on the lower right, where the mitochondria are stained red, and in normal healthy cells, they're distributed throughout the cells. But after treatment with terrestrials, they start to redistribute themselves, they circle the nucleus, and they change in shape and composition before the cells die. These compounds went on to further testing at the National Cancer Institute in their 60 tumour cell line panel, and they showed a degree of selectivity, enough so that they were advanced to an in vivo hollow fibre assay, but unfortunately they were inactivated by metabolism. This isn't an uncommon occurrence because it's much easier to kill cancer cells in a petri dish than it is in a living organism. Also done a lot of work on cyanobacteria since my postdoctoral days, and these are some of the families of natural products that we've looked at. Again, I'm just going to focus on one of them, and that's the toluporphins, of which toluporphin A is shown here. These compounds were isolated from a culture that was taken from a scraping on a stone building in Pompeii in Micronesia. They look, or the core of them might look very similar to a lot of you because it's very similar to that in common biological molecules that are called porphyrins. These porphyrins, however, and I've superimposed the core of the porphyrin on top here, they normally have a metal bound in the middle. In chlorophyll, for example, it's magnesium, and in hemoglobin, it's iron, but these compounds are always metal-free, and they also have this additional functionality on the core. Polyporphins turned out to be effective multi-drug resistance reversing agents. Cancer patients, after a while, become resistant to their chemotherapy because the cancer cells become resistant. They're actively able to pump a wide range of anti-cancer drugs out of the cell. It seems that the polyporphins block that pump and allow the anti-cancer agents to stay in the cells and do their work. These compounds are also excellent photosensitizers for photodynamic therapy. They preferentially concentrate in cancer cells, and then you can irradiate the cancer cell with laser light of a specific wavelength that activates these compounds, and they kill the cells. Quite a fun thing we did with them, but also with purpose, was to see if we could forcibly put a metal into the centre of these molecules, because they don't want to contain one. It was pretty hard work, and the only metals that they would pick up were copper 2+ ions and silver 2+ ions. And when they did pick up a metal, you got a very distinctive colour change from the metal-free purple to the metal-coordinated green compounds. We also tested both the metal and metal-free compounds, again for their bioactivity, and interestingly, there wasn't that much difference between them. Natural products from cyanobacteria are also implicated in water safety issues because a lot of them are toxic. And I've put 2 or 3 examples here: the microcysteines, nodularin, and anatoxin A. So-called algal blooms are actually cyanobacterial blooms, and they're a potential public health issue because if the toxins are present in those blooms, they may kill fish, dogs, stock, or even humans. And there have been some very nasty incidences where humans have been killed through drinking contaminated water. The picture on the lower right, I took that from the top of Tomato Peak in the Hawke's Bay, and you can see the blooms on these ponds, and you can also see a cow in one of the ponds. Not very good for its health. Natural products for water safety, so one of these are the microcysteines, as we've mentioned, and one of my PhD students, Jonathan Puddock, did a lot of work on these organisms. There are more than 270 types of microcysteine discovered, and all of them contain this unique amino acid called ADA, or some minor variation of it. We discovered 18 new variants here at Waikato, and Jonathan also did a lot of work on factors that influence their production, because again, it's very important to understand these things for monitoring and management. Natural products are also implicated in food safety issues. This is the work, this work was done with Cawthron with PhD student Sam Murray. This time he was looking at dinoflagellates. These are single-celled organisms. They're actually a type of phytoplankton. They can produce extremely toxic metabolites, and these metabolites increase in concentration as you move up the food chain, and there's a real risk of poisoning from consumption of contaminated fish or shellfish, and they have caused many deaths. So it's important to know the identity of these toxins, and again, which species produce them, so for monitoring and management issues. So one of the types of compounds produced by these tiny little organisms is this massive compound. This is mitotoxin. Other than biopolymers, mitotoxin is the largest known natural product. Not only that, if you want an example of how amazing nature is as a synthetic chemist, this is it. This compound contains 98 stereocenters. That means they're points in the molecule at which the arrangement in three-dimensional space can differ. If it's got 98 stereocenters, that means that there are 2 to the power of 98, and that's a 30-digit number, or if you like maths, 300 octillion, different forms possible of this molecule. Nature makes one specific one. It's just incredible. Anyway, Sam was able to isolate some new mitotoxins from one of his dinoflagellates. And he was able to determine most of the structure of this compound, which was quite a feat. So this is mitotoxin 7, and Sam used chemical transformations and mainly NMR spectroscopy to determine most of the differences between this compound and mitotoxin 1. So all the points in red are where he was able to determine an actual difference, and he knew that there was another methyl group somewhere in the blue part of the structure, and somewhere else in the structure there was another double bond. But to get this far is really remarkable. Another area of natural products that I really like to work on is chemical ecology. In other words, how do organisms use these natural products in their everyday lives? Not only does it provide us leads to potentially useful natural products, but if we can understand ecosystems better and how the organisms use the chemicals, again, it helps us to monitor and manage the environment better. This is one example of a chemical ecological project that we looked at. This work was done with the team who are listed there. And that's looking at, are invasive species more bioactive? One theory of how these so-called alien invaders can take hold is that they outcompete native organisms, and they do so with chemistry. So we looked at this both from a chemical and a biological perspective. I'm not really sure if we have the answer to this question yet, but the settlement plate shown on the right shows some of the interactions between some encrusting organisms that we observed. For example, these two organisms actually repel each other. There's a clear margin between them, so they're chemically fighting each other. So we can say that this is a tie. In the other interactions on this plate, there are winners and losers. We have one organism overgrowing another. What's interesting to me about this plate is that in 2 of those winning interactions, the organism involved is actually a native organism. It's a native bryozoan, and I really want to look at it, but we've got to get enough of it first to be able to study it, because this plate is actually very small. It's only 14 by 14 centimetres, so it's only a tiny amount of material there. Another chemical ecological area that I'm really passionate about is looking at the interactions between nudibranchs or sea slugs and marine invertebrate prey. These organisms are quite beautiful, as you can see, but they're also voracious carnivores. So they really like to eat marine invertebrates, but they themselves are soft-bodied and slow-moving, so they're also vulnerable to predation. So what they do is they sequester or take some of the natural products from their diet, and they use them either in their own defence or they pass them on to their eggs to protect them from being eaten. So if we can work out what the diet is and what the chemicals are, that both lets us discover new secondary metabolites and helps us determine the ecological role and source of them, and again, look at, are there any potential applications that we can use them for? I'm going to give you a couple of examples from my PhD student Loren's work. And the first is that looking at the nudibranch Goniobranchus oreomarginatus, or gold-rimmed nudibranch. These organisms were found on a black sponge, Dictyodendrilla, but they were found near a blue sponge, Decidia tiawanui. Loren collected all of these organisms and took them back to the lab and conducted food choice experiments where she offered the nudibranchs a choice of prey and then filmed hours of video to see what they did and then analysed the results. This is just a very quick snippet of the video where we see the nudibranch crawling on the black sponge. The results of this work were extremely clear. The nudibranch only wanted to eat the black sponge. In fact, they spent all of their time on it, which is an indicator that they're eating because they're grazing animals. So they only spent very small amount of time on the blue sponge. In fact, when you analyse the video closely, they just crawled through it and crawled away. They weren't interested. So of course, this led us to look more closely at the black sponge, and Lorraine was able to isolate 8 known natural products from the sponge, but more pleasingly, she was also able to isolate a couple of them from the nudibranchs as well, so this proved the predator-prey relationship. Further examination of the sponge led us to 5 new alkaloids, the denigrins, H to L. They were isolated from the sponge, as I said, but only in very small quantities. So we were only able to perform very limited biological studies on them, and they were done in conjunction with my colleague Linda Peters. So to try and get more of these compounds so that we can pursue them further, their synthesis is currently being examined by PhD student Xiaoming Liu under my colleague Ben Dixon's main supervision, but I'm on the supervisory panel too, so very interested in this project. Now, that was a very nice example where the results of the feeding experiments matched what we observed in the wild, and the chemistry backed it up. But unfortunately, these organisms aren't always that cooperative. This is another endemic species. This is called Aphlodorus luctuosa. And just as it swims around the tank slowly, I'll point out that it has antennae or feelers, or we call them rhinophores, on its head, and its gills are on its rear. In fact, nudibranch means exposed gill. So this organism was a source of a pyridinium salt. Lorraine was able to isolate it both from the nudibranch and from the nudibranch eggs, but we have no idea where the nudibranch gets this compound from. Despite looking at it a lot in the field and multiple collections, we were never able to observe it eating anything. It was just randomly crawling across sand. So therefore, we couldn't really conduct any food choice experiments on it because there was no potential prey to offer it. However, we know that similar compounds are exclusively found in sponges of the order Haplosclerida, and a couple of the species are listed there, Haliclona and Cleona. So we got a collection of a Haliclona sponge from relatively near the nudibranch collection site. And Lorraine and then third-year student Caitlin Bergquist extracted this sponge and analysed it. But unfortunately, they couldn't find any indication of this pyridinium salt. So then we asked the question, well, perhaps it's not dietary derived after all, or do we have the wrong species of sponge? Well, Lorraine was able to get further clues by unfortunately looking at the feces of the nudibranch. She looked at these feces, trying to see if they contained spicules. These are needle-like structures that are used in sponge support. And as I've hopefully indicated to you there were needle-like structures in the feces, so spicules were present, and the morphology or shape, in theory, might be able to tell us which sponge they came from, or in other words, what the nudibranch was eating. But unfortunately, in this case, the type of spicules present aren't very characteristic. 40% of all sponge spicules look like this. So we don't have a definitive answer yet, but Caitlin is continuing with this project as part of her master's research, and we're also hoping to get DNA analysis of the feces and or the gut contents to further tell us perhaps what the sponge is that the nudibranchs are eating. So the next few slides I'm going to be a bit vague and hand-waving because this material is commercially sensitive. But many of you will know that kiwi fruit is a very important industry in New Zealand. But that industry was all but decimated in 2010 when the bacterial disease PSA was found in an orchard in Te Puke. The industry's recovered to a good degree since then, but the current control methods aren't really satisfactory. Copper sprays are available and used, but they're phytotoxic, and the copper accumulates in the soil. Antibiotics are also used as a treatment, but you run the risk of the PSA evolving resistance to the antibiotics, so you can only use them in a limited fashion. There is a commercial spray available called Actigard, and that elicits the plant's natural defences, but it's only got limited efficacy, and it also has negative growth effects. So an alternative solution is required. So the question is, can marine natural products provide that solution? Well, we've already shown that the marine environment is an excellent source of bioactive compounds. And another plus is that terrestrial pathogens are unlikely to have had previous exposure to marine metabolites, so resistance is likely to be slower or less likely to develop. In this project, and it's a good team that works on these projects, Chris Battersill, myself, Joelle Venest from Plant and Food Research, and Mike Clearwater from the university, and then the students, whom I'll point out in a minute. So we've had 2 projects funded by Zespri thus far. The first was something of a pilot project, and that was the master's work of student Ashley Brown. And the results of that were promising, so that led to funding for a much larger project, and the students involved in that project were PhD student Lewis Dean and master's students Claire Voot and Taylor Farr. Again, the results of that were promising, so Joelle and I have expanded this project to look at a wider range of horticultural crops and a wider range of bacterial diseases. So far, we've had 2 projects in this area funded by a group called A Lighter Touch. That's a pan-sector group, which represents all the industries depicted there, and they're a part of Horticulture NZ. Again, results of the first project were very promising, so we've just received funding to go on plant. So we're now testing some of our compounds in on-plant trials, and should those trials be successful, the next step is larger field-based trials. So we're quite excited about this. Natural products aren't only useful in horticulture, they're also very useful in agriculture. This work was carried out with AgResearch with Sarah Munday-Finch and Alison Papay, and PhD students Singata Kauwhanonga and Nikki Webb. It involves looking at secondary metabolites of endophytic fungi, so fungi that grow inside the organism. Some of these fungi form a symbiotic relationship with common pasture grass, with ryegrass. And these fungi produce a whole range of secondary metabolites. Some of them are helpful because they prevent the plant from being eaten by insects, but some of them are detrimental because they cause disease in animals. So the idea behind this work is to look at novel endophyte strains, trying to find desirable properties, work out the structures of any novel metabolites that are present, and test those metabolites for anti-feedant activity against insects and also safety for grazing stock. So one example of the types of compounds that these endophytes produce are the epoxy xanthotrims, where epoxy xanthotrim 1 is shown here. These compounds are a feeding deterrent for Purina larvae. Purina is a type of moth, but unfortunately they cause ryegrass staggers in stock. That's a temporary neurological condition where the stock become uncoordinated and get tremors and stagger about. So they're not the perfect solution. So I can't say much about what Nikki's doing at the moment, other than the aims are to isolate and characterise new natural products from novel endophyte ryegrass associations and determine the effects of those metabolites on insect pests. And these pests include Argentine stem weevil, black beetle, and Purina, and then to assess animal safety. And of course, these new endophytes, the aim is to increase meat and milk production, and that's going to benefit the New Zealand economy. going sideways, and taking a bit of a different turn here, a project that we've recently started is looking at what makes plants of the Chatham Islands unique. This project is carried out in conjunction with Peter DeLong from Unitec, who's a botanist and a world expert on the flora of the Chatham Islands. It's also with Margaret Barber, and so far I have had 2 French interns work on it, Chloe and Gabrielle, and we've just had Jamie Lee, my new PhD student, start working on it. And this project is being carried out with the cooperation and the keen interest of the landowners on the Chatham Islands. Because the Chatham Islands have been isolated for so long, the plants have evolved uniquely, so they have a number of features that make them quite different from any counterparts they might have on the New Zealand mainland. For example, they're larger, they have more coloured flowers and fleshier leaves. This project aims to characterise the endemic species chemically and then compare that with any related species on the mainland to determine what it is chemically that makes these plants unique. And of course, the results are of great cultural significance to the Chatham Islanders. What about future work? Well, we've recently got some funding to look at marine natural products as safe and sustainable herbicides. There's a real need to replace these toxic and persistent herbicides, things like glyphosate and tordon, with things that are still effective but more environmentally friendly. Chris Battersill and I recently got some funding from MB to develop these herbicides from marine natural products. And our lead species for study have already been identified, so we've got a bit of a kickstart. The aim behind this work is to target the unique photosynthetic pathway of most common weed species. So if you only target that pathway, you're then not harming the species that you wish to grow. This work is being carried out in partnership with Ngāti Pukenga, who will be carrying out our field trials and helping with commercialisation. And there's also input from David Burrett from the University of Otago, who's a plant physiologist. Another area in which we're about to launch is looking at synthesising some more of our natural products so that we can test them further. This work is being carried out with Martin Banwell and Ping Lan at Jinan University in Guangdong, and with my colleague Linda Peters here for the biological testing. If you're interested in a bioactive natural product and you want to use it, sourcing it sustainably from the original source is not likely to be a viable means of obtaining it. We've already said that they're often produced in very small quantities. The production's highly variable, but also, of course, there are environmental concerns. You can't just go out and rape and pillage nature to suit your needs. So we need some alternative way of getting these compounds, and that's synthesis. Now, many of these compounds, as you've seen, are very challenging targets. But should the syntheses be successful, that allows us to explore their full biological potential through further testing. And of course, if we then find out that these compounds really are useful, that synthesis may prove to be a long-term source of supply. So I hope I have shown you how natural products, or nature's marvellous molecules, are both important to us and very relevant, and also fascinating. And I would now like to acknowledge my family. I would like to acknowledge my current and former colleagues, particularly all of my collaborators in this highly interdisciplinary work. And I would especially like to acknowledge my students, some of whom are shown here, but not all of them. They're shown here if I had a picture of them. They're the ones who do a lot of this fantastic work, and I'm very grateful to them. And I'd lastly like to thank you, the audience, for coming tonight. I really appreciate it, and for your attention. Thank you very much.
Tēnā koutou katoa. As Dean of the School of Science, so my name's Margaret Barber, those of you who don't know me, I'd just like to thank everyone for coming along tonight. I'll thank the staff, colleagues, whānau, guests, as we celebrate our newest science professor. So congratulations, Professor Michèle Prinsep. The School of Science, the university, and indeed, I'm not a chemist, but I think I'm going to say this anyway, the wider discipline of chemistry within New Zealand, thank you for your contributions. Michèle has been a stalwart of organic chemistry at this university for many years, and as Sarah Jane mentioned, is leading our chemistry and applied physics team for the school. Something that's always impressed me about Michèle is the way she elegantly balances academic excellence, which you've seen tonight, with a real passion and excitement for her chosen field. And I think you've seen that tonight too. That passion and excitement inspires the chemists of tomorrow. And those are high school students that she interacts with in all the many outreach activities she organises, and our own students who are lucky enough to be taught by Michèle. So not only is that excellence there, that passion and excitement is there, but she's also an incredible leader as far as empathy and kindness for the staff and students she interacts with. So all round, well-deserved and a stellar professor for the university. So we've heard tonight the wonderful, just a quick trip through some of the more exciting, interesting areas that Michèle's worked on over her career, and I was really struck by just how much real-world impact there is for each of these examples that she talked about. Not only anti-cancer, but saving our kiwi fruit crop, water safety, food safety. As a chemist, these are, I can only imagine how exciting it is to actually have real-world impact from the research you're doing, and Michèle's work does that. You know, I think a lot of us here are scientists, and if we're honest with ourselves, we'll probably admit how important publication metrics are. So I just wanted to impress you with just a tiny little snippet of Michèle's achievements. She has an H-index of 42, which is impressive, with over 11,000 citations to date, and that is truly impressive. Her field-wide citation index over the last 10 years, as far as I could tell, was 5.3. And those of you who are non-scientists, that means that Michèle's papers are noticed and used more than 5 times more than the average chemist, which is truly impressive. Personally, I'd just like to say that I was struck by how much cooler your organisms and your toys are than mine. A little bit jealous. You go diving and you see you collect and work on these most beautiful organisms. Then you bring them back. Okay, you put them through a blender. Perhaps not quite so cool, but SpongeBob had fun, I guess. And then you use super cool toys to measure what you're looking at. Yeah, I am jealous of that, and it's very, very impressive display of what an organic chemist can achieve in their research tonight. So that pretty much closes what we're the evening tonight. If you have any specific questions for Michèle, she'll be available in the foyer, and I think willing to answer questions. Yes. And yeah, so that concludes tonight's celebration of Michèle's achievement. And we look forward to seeing you at future professorial lectures. So thank you very much, everyone.